Yes, it was Wednesday, yeah, because we only had half a session there for lecture material. I want to go over neurotransmitters and neuromodulators again, just in a little bit of review, because classification of neurotransmitters, we talk about them as the classification. How are we going to get all these neurotransmitters classified? The chemical structure, how they're actually formulated, their effect on the postsynaptic membrane. Are we good with pre and post synapse here? That in the discussion questions in your lecture exam, I was really confused by some of you giving me some information on pre and post synaptic, and it was fairly evident that there was major confusion about what we talk about a pre and what we make reference to a post, and what happens at that synaptic cleft. Okay, so for some of you it was fairly obvious that there's some disconnect between what is pre and what is post synapse. We have a synapse, we have a junction there, and we have something that is at the end of the axon. We're talking about this stylized drawing I've been making over and over and over again like this, and we say here's the axon, and an action potential comes on down here. This is going to be the pre, and this is going to be post. Okay, so when we talk about a neurotransmitter, a neuromodulator is going to be released from a vesicle here. We're going to have to be released from a vesicle that is going to pass this cleft. This is the synaptic cleft. The problem is that calcium doesn't go across here. The neurotransmitter goes across there. And some of you were reporting that, well, the calcium has to be released here so that we can get opening of the receptors on the opposite side, on the other side of a synapse here. Well, the other side of the synapse is the postsynaptic portion of this. Okay? So calcium has to be entered in here to trigger the release of the vesicles here to release the neurotransmitter that has to diffuse across this. So we talk about what is the effect on the postsynaptic membrane. That's one of the other classifications that we have here for neurotransmitters. The mechanism of action at the target. What are two possible mechanisms of actions? What could they do?
Open ligand-gated channels.
Okay.
Can I close it? Or-
What's that?
Yeah,
please. Yeah, I've never experienced that. We had entertainment out in the quad out there at all, whatever you want to call it. Did we talk at all about inhibition or activation? A little bit of that, okay? Neurotransmitters can have an excitatory or an inhibitory effect on a variety of ways, okay? So they can either excite or they can inhibit, accelerate or brake. It's a very real issue associated with this. We talk about another possibility, ionotropic effect, which is binding to an ion channel. We also have metabotrophic effect here, binding to a G protein linked receptor. And we talked about that way back when we were making reference to something about the G protein had all those individual steps on it before we had that alylate cyclase, okay, the conversion. I'm not going to revisit that at this particular point here. Okay, the function of a particular neurotransmitter is determined by its receptor in the target cell. Okay, so we have to cross this. Neurotransmitter has to cross this, so there's a receptor here. There's something that has to receive that, and then it's going to be identified. It's going to be identifying something that's going to take place there. Okay, so some major chemical classes. Go back to here and we say the chemical structure. So let's look at some of the chemical classes that we have. And one of those that we've addressed numerous times here, we've talked about acetylcholine, best understood, most common. We make reference to some that are called biogenic. Anytime you hear gen in biology, you need to be thinking in terms of the genesis of it, in the beginning of this biological formation of it. And I made reference to some which we said were catechol, and here it's biogenic amines. Well, here's the catchphrase that you want to just attach to here. catecholamines, indolamines, okay? And that's gonna give you a clue that says, well, this must be one of those neurotransmitters that's related to those amines called biogenic amines. Now, you also should get real comfortable with one called norepinephrine, serotonin, dopamine. Those are ones that are gonna come up. Boy, if you're gonna go into any identification with pharmacology at all, if you're gonna go into nursing and you take a pharmacology class, you're probably gonna hear about dopamine. And you're gonna hear about epinephrine and norepinephrine. You're gonna hear about serotonin blockers, okay, some things such as that. So those are gonna be real common terms in a vocabulary of a person who's in administering drugs, doing anything with medications with patients. All right, so have we talked about amino acids? Geez, I think we beat the crap out of those on time or two here. We talk about amino acids linked together. You get 50 or more and you end up with a protein, but you don't always have to have a protein. You can have a polypeptide. You can have a link of, or we can talk about something called a neuropeptide. Gamma-aminobutyric acid, glycine, and glutamate. The glutamate, you may understand, have you ever heard of monosodium glutamate? Ever heard about Asian food? Oftentimes they say we use a tenderizing agent there, monosodium glutamate. Okay, well there's glutamate as one of the amino acids associated with this. Purines, pyrimidines, that's something that you may have come across in some of the bio classes here. Nitrogen-containing compounds derived from nucleic acids. Have you heard of nucleic acids before? You should know too, right? And you were going to tell me they're what?
Deoxyribonucleic acid and ribonucleic acid.
You got it. And you shouldn't be deer in the headlights when you hear the word nucleic acids, but oftentimes bring it up to a group of people and say, geez, never heard of those before. Yes, you have. Examples include adenosine. Have we talked about ATP at all? Okay, purines here. All right, so ATP can even be acting as a neurotransmitter, oddly enough. So some of these have multiple behaviors or applications. Neuropeptides. Here is, when you hear the term peptide, you have to think, well, that peptide bonds are where you link some amino acids together. You put enough of them together, you have a protein, but you don't always have to have a protein when you put amino acids together. You can have something called a neuropeptide, a short chain of amino acids. Less than 50 probably, more than 10 oftentimes we consider. And it says include substance P. And we have addressed a little bit of a term called an endorphin. Endorphin is a substance that's considered to be a product produced by the brain that is a pain-suppressing type of, has a pain-suppressing type of activity. We have some gases and lipids that also can act as neurotransmitters, include gasotransmitters, nitric oxide gas. Nitric oxide, you need to write down, that's a vasodilator. It's a major vasodilator. Nitric oxide. Major vasodilator. We talk about carbon monoxide. Carbon monoxide, odorless, colorless, tasteless, poisonous. Okay? Gas. And endocannabinoids. Have you heard of endocannabinoids? Someone want to give me an example of an endocannabinoid? Weed. Right? Okay. So this table comes from your text where it kind of busts them out. Neurotransmitters, acetylcholine, biogenic amines, amino acids. Oops, jumped ahead too far. Purines, neuropeptides, gases and lipids. No, it's just covering this again. The endorphins here are widespread in the central nervous system. We notice this. What's the effect on the target tissue here? Inhibitory. We want you to have some feel-good feelings here, okay? Endorphins reduce pain by blocking the release of substance P, produce a feeling of euphoria. We talk about people who get the endorphin rush. Aerobic exercise is oftentimes attributed to the release of endorphins. We go over here to, I mentioned about norepinephrine. Some of the central nervous system synapses, it's generally considered to be excitatory. What is this ANS synapses? that for?
Autonomic nervous system
Which is what?
is the,
Yes?
like the parts of it,
Yes?
sympathetic and parasympathetic.
Yeah, sympathetic, parasympathetic, okay? So he's talking about, notice here, here's dopamine, ANS synapses. Here's ANS sympathetic, most sympathetic targets, norepinephrine, most of the sympathetic targets, and it's excitatory. What does that mean, a sympathetic target? Fight, flight, or flight, right? Amphetamines increase extracellular norepinephrine and dopamine levels by blocking the reuptake, which leads to a euphoria situation. We hear about people, drug trade of, before fentanyl and stuff, that amphetamines were a big deal on the streets, okay? So there's all kinds of things here that you can look at with regard to, how they're clinically connected, what's the target effect here, the other things that go along with these. Okay, responses at the postsynaptic cell. Are we good with this, postsynaptic? If this is our axon and we're trying to get a signal to this, muscle, gland, whatever is down here, this is the action potential has to cross the synapse. And we have this here, he says, response is excitatory and inhibitory postsynaptic potential. Excitatory postsynaptic potential, EPSPs. You have to know about a little bit of information about EPSPs. Notice what this is going to be doing right here. Here's our minus 70 millivolts, okay? And for that testing purposes, a number of you just put 70, and you didn't tell me what 70 meant. Is 70 the temperature? Is 70 the number of people? You said we have to get to 70. 70 what? What does 70 represent? We have to get to 50. What does 50 represent? Does anybody remember what 50 represented? 50 what? Millivolts. You could have just put MV there and that would have been good. If you don't put MV there, I don't know what you're talking about. We started with 70. And then we had to get to 50 and it went up to 20. Where are you going? Is that the speed limit? Are you following what I'm talking about? No identification of terms behind it, and it means nothing. Well, in this case here, we've used 50 as threshold, didn't we? We had to get to 50 before depolarization takes place. What the heck is 50? Okay, notice this, excitatory postsynaptic potential. Here's our timeline. Here's our millivolt value right here. Resting membrane potential. The resting membrane potential is maintained by the sodium-potassium pump. Okay, depolarization occurs in response to depolarization occurs and the response is going to be stimulatory. Stimulatory says, go. Depolarization might reach threshold, producing an action potential, and the cell is going to respond, right? There'll be a cell response there. Inhibitory postsynaptic potential, hyperpolarization, and the response is inhibitory. Do you recall what the whole deal was with hyperpolarization? Resting membrane was what?
70 to 90.
Minus 70
70.
millivolts.
So millivolts.
So what was hyperpolarization?
Under minus 70 millivolts.
Yeah, even more negative. Okay, which means that if this is minus 70, in order to overcome this, we have to have a stimulus that is great enough to get us to threshold, but it has to be super strong because we have to get that hyperpolarization back to at least minus 70 millivolts. Okay, so an inhibitory postsynaptic potential is one that can create a hyperpolarization inhibitory. It decreases, the hyperpolarization decreases the likelihood of an action potential by moving the membrane potential farther from threshold. It's even more electronegative. So it's just very difficult for that to take place. Okay, so this whole topic on this last section here, at least this portion of it, was related to neurotransmitters and neuromodulators. Modulators are something that you say can dial it up or dial it down. Okay, we can dial up the activity or dial it down. We made reference to, here is the our myelinated axon. Here's our here's our post, presynaptic neuron, and it has the vesicles in it. The action potential comes on down. But there is another synapse between this inhibitory neuron and this presynaptic neuron right here. And in this instance right here, where we can have a neuromodulator, can influence the likelihood of an action potential being produced in the postsynaptic cell, because we influence the release of neurotransmitter here that's going to cross this. And this one, they're showing you, hey, that there's a dumping of the neurotransmitter in the synaptic cleft here that is capable of being picked up by receptors on the postsynaptic membrane here. However, in this case right here, what we're seeing is some kind of influence by this particular inhibitory neuron that's going to modulate that. It's going to influence the release of neurotransmitter into the synaptic cleft. And what the author here of this illustration is trying to demonstrate, he's got a bucket of the neurotransmitter that's been deposited in that cleft, but over here, dramatically less, which may be interpreted then as a reduced amount of activity that can take place in this postsynaptic membrane. Okay, so axoaxonic synapse. Does that make sense? Decode axoaxonic. We're having a synapse between what? Two axons. This axon and that axon. This one, the presynaptic and the inhibitory neuron. Axon of one neuron is going to synapse with the presynaptic terminal axon and number, and that is reported to be fairly common in the central nervous system, where we can influence that. For any of you who are going into nursing, you're going to be dealing with pharmacology. You're going to be dealing with how the drug is influencing the release of a neurotransmitter, the uptake or the inhibition of it at these particular locations right here. Presynaptic inhibition, reduction in the amount of neurotransmitter released from the presynaptic terminal. An endorphin. Did we just talk about an endorphin? It can suppress that release, so you can have an inhibitory effect here, can inhibit the pain sensation, give you that euphoria. Presynaptic facilitation, the amount of neurotransmitter released from the presynaptic terminal increases. Glutamate is one of those, facilitating nitric oxide production. What did he say nitric oxide was?
The vasodilator.
Vasodilator. Okay, the rest of this information is just talking about how we summate. Does everybody recall what summate is? Where we're going to add things together, okay? We're going to put things together. A single postsynaptic potential is not sufficient to reach threshold, okay? We're good at that? A single is not capable of reaching threshold. So what do we have to do? What's the answer here? What do you have to do? You gotta get, come on buddies, we gotta get together here and get this going, okay? Gotta bring your friends. Most postsynaptic potentials, a single postsynaptic potential is not sufficient. Many postsynaptic potentials combine in summation at the trigger zone if threshold is reached. Remember, if we reach threshold, remember it was an all or none, correct? We get to threshold, it's all or none from that point. Two types. We have spatial and we have temporal summation, putting things together in this manner. So the authors of this piece put together something like this. They say, okay, here's spatial summation. Action potential 1, down, okay? And 2, okay? Cause the production of graded potentials. Remember what graded potential was? Take a little stone and you throw it in here, and you got another little stone and you throw it here, and the two together combine so that you have a little bit more of a wave pattern that comes out from the stones hitting the water, okay? At two different dendrites, remember dendrites, cell body, axon, dendrite, cell body, axon, okay? These graded potentials add together at the trigger zone to produce a graded potential that exceeds what? It exceeds our -50, our -50, okay? It's gone from -70, resulting in, bam, all or nothing, action potential. That's the way you should look at that and be able to interpret and just say, oh, I got it. We're going to put these together, we're going to add these two together, and they're going to reach that -50, and then once it reaches that threshold, it gets from -70 to a -50 millivolts, then we're going to have this thing trigger the action potential. Temporal summation. The temporal summation now, two action potentials arrive in close succession. Boom, boom. Okay, rather than boom over here and one over here and they summate, this is going to be boom, boom. one on top of the other, so to speak. The FERT, okay, the first action potential causes the production of a graded potential that does not reach threshold at the trigger zone. The second action potential results in the production of a second graded potential that summate reach threshold, action potential. Okay, combining these, we say temporal with both EPSPs and IPSPs. Okay, are we good with EPSPs? You have to understand what the E stands for there. E is all about what? Accelerator, okay, it's excitatory. Hit the gas. The IPSP here is inhibitory. It just says, okay, so what we're dealing with in this case here, we're gonna start right here. This is an excitatory event, here's an inhibitory event, here's an excitatory event, inhibitory event, here's an excitatory event. Okay, we're gonna see, are we gonna generate an action potential? What do we have here? An action potential is produced at the trigger zone when the graded potential is produced as a result of EPSPs and IPSPs summate to reach threshold. One of them has to exceed the capability of the other. The summation of the EPSPs have to exceed the IPSPs in order for us to be able to actually have something that reaches threshold here. All right, this, we're not gonna go into reverberating parallel after, okay. Are there questions? That's a lot of information. Are there some questions relevant to that needed to come up? Okay. Move on. Okay, I gave you a sheet that I want you to fill out here. And it's this one right here. Okay. All the answers are on the back. That's the main thing. I'd like to go over it with you here. First of all, the top bracket up here, you should know how to do this. This should be something that you take a look at and you say, oh, I got the answers, Dad. Number one should be nerve system. We're going to look at the nerve system in total, a big picture nerve system. Number two, you should have over the central nerve system. It's going to be the CNS. CNS is number two. What should number three be then? Peripheral nerve system. Yeah, and the bracketing there below, you should have PNS. Now, notice the arrow going between central nerve system and the peripheral nerve system. What does that mean? What do you think that means? Really? Come on, guys. There's an arrow between number two and number three. You see that? Arrows there, what might that indicate?
That they work together?
They communicate together, right? Yeah, that sounds like a brilliant idea. Why don't we have the central nerve system, peripheral nerve system talk to each other? Okay, number four. Under central nerve system, remember there were two components of the central nerve system. Number four should be brain. And then number six down there, we should have spinal cord. So four and then drop down to number six. So brain and spinal cord are those components of what we'd say is the central nerve system, okay? All right, so let's go over to number five. Under peripheral nerve system, there's that lines going down, linking up number five to number three. How about cranial nerves? And in that bracket, how about cranial nerve number one through cranial nerve number, how many do we have? 12, yeah. Cranial nerve number one, Roman numeral 1 to cranial nerve 12. Okay, so under peripheral nerve system, we're seeing that we have the peripheral nerve system is the cranial nerves. Well, number 7 should be what? If we have cranial nerves identified in one bracket, what should we have in the next bracket there? How about the spinal nerves, okay? So you have the cranial nerves and the spinal nerves. Now, under spinal nerves, we're going to take out those 4 pieces there, 8, 9, 10, and 11, and we're going to have number 8 as cervical nerves. Now, how many cervical vertebral segments do we have? Does anybody recall? 7, but we actually have 8 cervical spinal nerves. So you should have in that bracket there under number 8, you should C1 through C8. cervical spinal nerve number 1 through cervical spinal nerve root number 8. And there's a right and a left component to those. How about number 9? We're under spinal nerves. Cervical, what would be next, logically?
Thoracic.
Thoracic, right? Thoracic. Thoracic nerves. How many of those do we have?
12.
Yeah, T1 through T12. You have those nerve roots T1 through T12. So you have cervical is 8, thoracic is number 9. Number 10, well, we're moving cervical, thoracic. What's the next in line here? Lumbar. Lumbar nerves. We have the lumbar nerve roots. We have those that extend to the right roots and roots that extend to the left, and that's L1 through L5. And then number 11 down there is going to be the sacral nerves. And we're going to have sacral nerve S1 through S5 in that case. Didn't we do this before? Okay, down on the bottom, on the left-hand side of the page there, number 12, why don't you put in just those 3 letters C and S, and number 13, put in P and S. And then you have, there's, let me get the arrow right here. The sensory afferent is, you know, you got an arrow going this way, and the motor efferent is going this way. I think those arrows are going the wrong direction, aren't they? Shouldn't the CNS be, shouldn't the motor efferent go from the central nervous system to the peripheral nervous system? Are those arrows correctly directed? You gotta help me out, guys. The arrows are on the page there. Which way should sensory be going? Should be going which direction? From the periphery to the central nervous system, right? Aren't those arrows reversed?
They go backwards.
Aren't those arrows backwards? You gotta help me out here. Does this sound logical at all? Okay, I just put the arrows on here like this, and as we're doing this right now, wait a minute, that doesn't look right. It appears that the sensory afferent should be going from, because this is CNS over here, and this is PNS over here. So we should have this sensory material going to the central nervous system, and the motor afferent information should be going in this direction to tell what to do. Remember, the motor afferent is saying, hey, do this. You gave us this information here, this is what you should do with it. So it's my interpretation that that would be inaccurate to have it as it is listed right here. Okay. You should be able to do this. I mean, this is something you should take a look at this real quick and say, oh yeah, brain and spinal cord, spinal nerves, cranial nerves. Okay, when you look at this material right here, it should be kind of fall in place in that particular manner. Just let me get us into, we're gonna go into a little bit of 12. We're not covering all the material in 12, but we're gonna cover a number of pages of that information, and then we'll drop into 13. All right, so the spinal cord extends from the brain at the level of the foramen magnum. We're good with foramen magnum? Okay, from the level of the foramen magnum down to the level of the second lumbar vertebral segment. Second lumbar. If you put your hands on the crest of the ilium right here, and you go in, you're about 4 or 5. You're right at the junction kind of L4, L5 right here. So where would L2 be? If this is 4, 3, 2. Your second lumbar vertebral segment is going to be someplace right in here, okay? That's the end of the, if you follow this illustration right here, here's the end of the, what we call the spinal cord, okay? It ends at the level of the second lumbar vertebral segment. Doesn't extend, the spinal cord doesn't go all the way down to the coccyx area right here. It's up there a few segments, lumbar segments. So the spinal cord is composed of segments, each associated with a pair of spinal nerves. We got that pair, we already went over that. We have, how many in the cervical?
I mean, seven. Seven.
Really,
Went over it, I even emphasized.
You want to see?
What was it? How many in the cervical?
8.
Eight.
Eight pair,
Oh, you're talking about the nerves.
7 cervical segments, 8 cervical spinal nerves, okay? We have 8 there. What about in the thoracic?
12.
Now there's gonna be 1 for each segment there. In the lumbar, we have 5. In the sacrum, oddly enough, we actually have 5. S1 through S5, okay? So the spinal cord then is composed of segments, each associated with a pair of spinal nerves. And you see they're going from each direction off of this spinal cord. Now, one of the other things we're gonna have here, segments are identified according to the portion of the vertebral column from which these nerves enter and exit. We're gonna make reference to some enlargements that we see associated with this. So, the spinal cord extends from foramen magnum and second lumbar. Segment, there's a cervical, there's a thoracic, there's a lumbar, and there's a sacral section associated with this. Okay, and we see your spinal nerves extending out. The cord is here. We have the vertebral column and we have the spinal cord. So it gives rise to 31 pair. So what we're actually looking at is 31 pair of these spinal nerves that are extending away from the cord here. Cervical, thoracic, lumbar, and sacral. Not uniform in diameter throughout the length. We have a cervical enlargement. We see in the area here, there's a little bit, then it thins out, to the extent of the thoracic, and then we're gonna have down in here the lumbar enlargement supplying the lower limbs. To a certain extent, that makes some sense. We have the area in here, in the thoracic area, does not have the need for, the extent of the nerve communication out to the upper or lower extremities, which is then provided by these enlargements right here. A term which we're going to talk about in detail, and again, I mentioned to you about having your lab manual, because some of this is going to make some sense. Where we talk about the conus medullaris. The conus medullaris is this tapered end right here. And then we have this area right here, which the cauda equina, and any of you associated with horses, equine is generally made reference to horses here, and so the equine, or the mare's tail, is this portion right here. Cauda equina is the tail of this, which then fans out, and we have all of these fine little hairs of nerve that extend away from the conus medullaris here. And then we have, and this identified as right around the second lumbar vertebral segment, right in here. And then we have this fine, thin little strand that almost seems to be like any of you have ever gone out camping and you have maybe a rope that you have to tie down the edge of your tent or something like that, and you stake it down with a little peg down at the end. It always gives me the impression that is this little tight wire that you're pulling there to keep everything kind of stretched tight along that entire area, and we say that this is the phylum terminale. All right, so we have here the conus medullaris tapered at the inferior end, the cauda equina, origins of the spinal nerves extending inferiorly from the lumbosacral enlargement, and then the conus medullaris. We have all of that mare's tail type of thing. All right, when we get into the lab, our next lab session, which is 2 weeks, this week we have this Monday we have the exam, and then the following week we're going to be dealing with nerve involvement and then reflexes following that. We're going to be dealing with a lot of this material here where we talked about the meninges. So we're going to address it a little bit right now, but we're going to actually do a saturation in the lab there, where you talk about the dura mater, the tough mother, okay, dura mater continuous with the epineurium of the spinal nerves. You also might hear people call it the thecal sac. Then there's the arachnoid mater, okay, and any of you who are, anybody here arachnophobic? Which means what? What is that?
Spiders. Fear of spiders.
Fear of spiders, okay. So you have arachnophobia. Okay, so arachnoid mater is going to be a spider webby layer. When we do the dissection of the sheep brains here, you're gonna be able to see dura mater. I've never yet identified the arachnoid mater. And the reason I think that that might be the case, the artist is interpreting it right here as these little fine little lines that are in this area right here. It's like spiderwebby layer of connection within that area between the dura and the next layer down to the pia. But the arachnoid layer right here, and it may be because of the preservative that's being used, or it's very wispy. If you probably had a fresh brain to look at, but ours have been swimming in preservative for quite some time there, so probably don't see it. But they say thin and wispy right here. And then we have the pia. So we have DAP, if you want to get a real quick reference to that. DAP, dura arachnoid pia, bound tightly to the surface of the brain and the spinal cord. And the pia, when we look at the brains in the lab there, it's going to be looking like a little bit of a saran wrap glistening material that's right over the surface of the cortex of the brain. The pia also said to form that phylum terminale. Remember what the phylum terminale was? Okay. Phylum terminale, that little thin wisp of tether material here. It almost seems like it's something that's pulling tight to hold it in place here. And the denticulate ligament that attached the spinal cord to the dura here. All right, spaces. We're going to find that these are going to be really important for any of you who are going to be in the healthcare field there, and you have to do epidural, or you have to do some type of spinal tap, or you're involved with the procedures there. You're going to find that epidural should give you an indication. What does that lead you to think in terms of decoding that word?
Epi at the top,
Above the door.
above the door.
Above the dura. Yes, that's accurate. Location of epidural anesthesia. Person gets an epidural, they're getting where? Well, it's going to be not into the dura, but it's going to be on this lining the dura, so it's going to be to that point here. It contains blood vessels. Here's that areolar connective tissue, but there's also some fat based in that area. So we have epidural. Then we have subdural. Where do you go for a subdural? Well, you're going to have to penetrate through the dura. Okay? You're going to go through the dura in order to get to that. And then you had subarachnoid. Okay, now I mentioned to you that that arachnoid was this real wispy area right here. So if you're subarachnoid, if this is dura, and then this layer right here is the arachnoid mater, and then you get deep to that before you get to the pia mater, subarachnoid, we find that CSF and blood vessels within the web-like strands of the arachnoid tissue. When they do fluid out of that area for analysis or evaluation, they're going into that subarachnoid space there to withdraw some cerebral spinal fluid. That's the cushioning fluid there. We want to find out we talk about, these are meninges. What would an infection of the meninges be referred to as?
Meningitis?
Meningitis, right? Itis is inflammation, meningitis. So two concerns we always have, if a person is manifesting some symptoms associated with that, is it viral or is it bacterial? Is it viral meningitis or is it bacterial meningitis? Both of those can be, have some serious consequences associated with them, but they'll be doing an analysis with a spinal tap type of thing. All right, so let's put this into perspective. Here we've got this massive drawing of the spinal cord, okay? Now let's put it into proper perspective. Here's where it's sitting inside this opening right here. What's that opening going to be referred to?
Vertebral foramen.
Say it again, please.
Vertebral foramen.
Vertebral foramen. Remember a hole, or the hole in the bone? It's in the vertebral foramen right there. So all of that tissue that we're making reference to right there, and a bunch more because they've added a whole bunch of fat in this area right here. They got epidural space filled with adipose tissue. So this is cushioned with some fat around it, but this is that space here. Here's our dura mater. Okay, arachnoid, pia mater is and here is the spinal cord in cross section. And we see this kind of interesting, they always refer to it as a butterfly pattern in here. Well, you're going to have to know the features of the butterfly pattern. It's going to be important for you to appreciate that. Here's the spinous process, our transverse processes, our body of the vertebral segment, and this is the vertebral foramen. These are going to be the intervertebral foramina here. If there was a bone above, and we got this bone right here, then we're gonna have a potential space there, only because there's a bone above and a bone below. Here's our spinal nerve extending out, okay, if we're looking at it from above down, right side, left side, okay? All right, so we're gonna see then a few features of this. Couple of terms that are gonna really, really be important for you to nail down. Ventral and dorsal, okay? Are we good with ventral? Ventral is out here and dorsal is back there, okay? And so we're gonna be talking a lot about a ventral root and a dorsal root. And one of the other key features that you really need to nail down is this dorsal root ganglion, okay? And the ventral root and the dorsal root are actually gonna confluence here, so we're gonna end up with this being the spinal nerve. Two components of it. This is coming off of, this ventral root is coming off of the ventral surface of the spinal cord, and the dorsal is the posterior aspect right here. So we're going to talk specifically about what's going on with dorsal and ventral here. So anterior median fissure and posterior median sulcus. What the heck does that mean? A sulcus is a depression like here, a fissure or something like that. We've got, if you're a person who sulks, have you ever heard that term used? Why are you sulking? What might that make reference to?
Sinking in.
I'm sorry?
Sinking in.
Sulk. Sulking. You just kind of sulking. Just woe is me. Okay? Sulking. Sulk. A sulcus is a depression. Okay, so think in terms of that. When you hear the term, we're going to use it a lot when we get to talking about surfaces on the brain, the cortical area specifically. Anterior median fissure. Fissure is going to be a crease in there. A sulcus is going to be a depression there. Deep clefts, okay, partially separating right and left halves. Are we good with white matter? Made reference to that before. When it's white matter, it's said to be myelinated. What was the deal with myelin?
They help conduct electrical signals.
It's an insulator, right? Helps speed up the ability for communication of a signal, right? It's going to be really important. Myelinated axons forming tracts. Now, we're in the cord, and here's a real important term for you to nail down early on here. When we make reference to a tract is within the spinal cord itself. Okay, tracts. Nerves outside, tracts within the cord. Okay, 3 columns. They're also referred to as funiculi. There are 3 columns or funiculi. We're going to have a ventral. Where would you look for a ventral column? Yeah, toward the front. Where would you look for a dorsal? Toward the back. Where would you look for the lateral? To the side. Let's go back to this illustration right here. This is the cord. This area back here is going to be the columns that we're going to refer to as the dorsal. These are going to be ventral here. These are going to be the lateral over here. Okay, real simple. Dorsal, ventral, lateral. Lateral columns there. Ventral columns, dorsal columns. Okay, each of these divide into tracts, pathways. The tracts are pathways. The gray matter is what? Unmyelinated, right? Slower response here. Dendrites and axons, neuron cell bodies, dendrites and axons are unmyelinated, the gray matter. All right, we're going to talk about posterior, dorsal, anterior, ventral, lateral associated with autonomic nerves, and those are the horns that we talked about here. We're going to deal with terms called commissures, connections between left and right. You have to communicate between this side and this side. You're going to have something that's communicating from one side to the other. And then the white here, we're going to gray and white commissures. Roots. We've already talked about spinal nerve arise as rootlets and then combine to form these roots. Rootlets, then the roots here, okay? Dorsal root. Okay, nail this down right now. What information is being communicated in a dorsal root?
Sensory.
It's right here. What is it?
Sensory.
Sensory. The ventral root? Motor, okay? So the ventral root is going to be our motor communication. Which direction is motor communication traveling? From the CNS to the PNS, right? Okay, which direction is the sensory information going? From the PNS to the CNS, okay? Please tie that down real quick. Put a, peg on that thing real quick here. Dorsal, sensory, ventral, motor. Okay, what can we have up here? Sensory afferent, what would we put up here for sensory? Sensory is gonna be which one?
Dorsal.
This is gonna be the dorsal root associated with it. What about the motor? Ventral. Okay, so if you wanna link those things, that'd be a real good idea to get that nailed down real tight, real early, because anytime we're talking about which way is the communication going, which way is that signal going? Well, if it's dorsal information, we're sending it up for analysis. I wanna find out, well, what is this important or not important? And then when it gets up there, the analysis is taking place and it says, yeah, do this with that. Get your hand off that burning piece of wood. Okay? All right. The two roots merge laterally to form a spinal nerve. Are we good with a lateral merging? Just mentioned we have these two. We have a dorsal and a ventral, and they're going to come off of the spinal cord here, and then they're going to merge, and then they're going to form one. Okay? We'll go back to that, and we'll see it here again in this illustration. Ventral and dorsal. Which one is carrying the sensory? Which one is sensory?
Dorsal.
Dorsal, right here. So this dorsal root ganglion, that's carrying the sensory information, and the ventral is carrying the motor. Now here are the rootlets, and then we're going to form the spinal nerve as we get laterally. As we laterally go through this foramina right here and out to the extent along the body here. So the dorsal is carrying the sensory information, the ventral is saying, hey, do this with that information. The two roots merge laterally to form a spinal nerve. Does that make sense? That one comment right here. The dorsal and the ventral are going to merge as we go laterally to go out to the periphery of the body. Okay, dorsal root has a ganglion. The ganglion was that wide spot in the road. Okay, and that's what we saw in that illustration. Here we see it again. Here's the dorsal root ganglion. These are what we make reference to as the areas here. Here's our gray matter, here's our white matter around here. Okay, the white matter is posterior, lateral, anterior. We're also going to have posterior, lateral, and anterior on the area called the commissures here. This commissure is communicating between here and here. Between this side and this side, we have to have a commissure here. This is what it actually looks like, photomicrograph of this spinal. That's not even a micrograph. It's a, we're going to see some spinal cord, cheap spinal cords that are cut, and you can see this pattern right in a cut section of the spinal cord there. Okay, and now here in this illustration in your notes here, you're going to see something that's kind of interesting. We have ascending. These text authors couldn't go with the convention everybody else uses red and blue. Okay, these guys use whatever that is and whatever that is. Sea foam and lavender, I guess. I don't know what they're going to call it. Okay, but notice there's arrows specific. We got all of this green right over here. Ascending nerve tracts. What are the ascending tracts carrying?
Sensory information.
Please. What are the ascending tracts carrying? Please.
Sensory.
What is it?
Sensory.
Yes, sensory. They're carrying sensory information. And notice the artist here is trying to let you know that the ascending are going up and the descending are going down. They're called tracts. This lavender right here. Now notice, do you see one is more dominant than the other? What looks to be more dominant there?
Ascending.
The ascending. Ascending meaning what?
Sensory.
Sensory. And that is generally considered to be the consensus by most neuroanatomists. They say we have 2/3 sensory, 1/3 motor. What does that tell you? What might that lead you to think in terms of? Spill something. What is it?
Constantly sending signals up to the brain whenever we feel or touch or taste something. Right.
So what is all of that information supposed to, what are you supposed to do with all that information? Is it all important?
Process of the brain.
No, there's a whole lot of stuff going on in the room right here. In the background, you might be hearing some noises or people shuffling around. So is that important? No, it's not important. But there are some things that you might consider important, okay? And so what it amounts to is we get all of this sensory information. And I mentioned to you a couple of different times, I told you you have a, we're gonna learn about a portion of the brain called the thalamus. And the thalamus is what I always call the crap finder, okay? It's out there and it's a screen, and what's it doing? It's kind of shuffling through all that information that's coming in. 2/3 of the information coming up is sensory, but we're only sending a signal out relevant to 1/3 of the information that we get. So there has to be a whole lot of superfluous information there that we get that we don't necessarily have to act on it, but it's there in the event that we do have to act on it. You follow what I'm talking about? And so these ascending tracks, there's a whole lot more of the ascending tracks than there are the descending motor tracks. And what it's telling us is that there's a lot of information there. Maybe this would be important at some point, but maybe not right now. And so the thalamus is this screen. It takes all that information, and it's like this wizard that's up there, and all this important, not important, not important, not important, important, not important, not important, not important, not important. You follow what I'm talking about? And it's doing that all day. Probably more so when you're awake than when you're asleep, but there's also a lot of processes going on physiologically in your body while you're asleep that have to be screened as well. Breathe. Don't breathe. Breathe. Don't breathe. Don't urinate right now. This is not a good time. You're in bed. Nope. Okay? Do you follow what I'm talking about? There's a whole lot of screening that has to be done, and that's what's going on in this information up, information down. These are tracks. These aren't nerves. Once we get out here, these are the nerves. These are the ascending and descending tracks. Sensory, motor tracks. Ascending tracks are sensory. Descending tracks are motor, passing along the cord here. And notice where they're located. They're located in this area. Is that the white matter or the gray matter? What is this area out here identified as?
The white matter.
White. Dorsal, ventral, lateral. White. Dorsal column, ventral column, lateral column of the cord. Those are said to be demyelinated. Why myelinated?
Faster processing.
It's supposed to be a little bit more rapid communication capability there. Okay? Then we have gray matter, the dorsal, lateral, and ventral within this butterfly pattern right here. All right. So we talk about the relationship of the sensory and motor neurons in the spinal communication right here. All right. This area right here is carrying what kind of information? Sensory. It's a dorsal spinal nerve right here, sensory neuron. Sensory. It comes in here, and then it's going to communicate with an inner neuron within the gray matter here, the horn, and then it's going to be passing out. We talk about autonomic neuron and a somatic motor neuron here. Okay, autonomic motor neuron and somatic. So let's see what goes on. Dorsal ganglion, collections of cell bodies of pseudounipolar. We have to have that too. Okay, sensory neurons forming these enlargements, these kind of bubble-like deals on this dorsal root back here, posterior, and we say that those are the dorsal root ganglion. The motor neuron cell bodies are in the anterior and lateral horns of the spinal cord. The anterior and the lateral, okay? So we're going to find here that we have multipolar. What was the most common type of neuron we talked about?
Acetylcholine.
Wasn't it the multipolar? Oh, so quickly forget here, come on. Anterior motor horn here. Autonomic neurons in the lateral horns. The autonomic, when you talk about sympathetic and parasympathetic. Axons of motor neurons form the ventral roots, okay, and then they're going to pass out to the through the spinal nerves out to a particular area of the body. So this information is gonna be carried over in the lab that we're gonna do. We have a lab, entire lab that's just related to reflexes. Okay, so we're gonna talk about an automatic response to a stimulus that occurs without conscious thought. You blink when you just blink. How can you blink?
My eyes are dry.
Yeah. So it's a response to, I gotta wet the surface of the cornea, right? Gotta wash the surface of the cornea. And so you blink. Is that, can you not blink? For a period of time you can, you just, I'm keeping my eyes open, I'm not gonna blink, I'm not gonna blink, I'm not gonna blink, I'm not. But then at some point, you know, you get this, and you blink, okay? All right, homeostasis. We're gonna make reference to something called a reflex arc. It's a neural pathway that controls the reflex. You need to know these 5 components of the reflex arc. They're right here. There's 5 components. What might be the first one that you have to have? How about something that even tells you that something's going on? You need to have a receptor, something that's a sensory receptor. On your automobile, it's a little deal that it says, hey, you get too close to the car in front of you, right? Or there's a car coming up alongside you. A little sensory thing does that. And what does it do? You have a receptor, which is attached to a what? A neuron. What does the neuron do? It goes to an interneuron. What's the interneuron all about? Well, it has to communicate to a motor neuron. What is the motor neuron? It says, sends a signal to something, says, do this. Okay? These are the 5 steps of that you need to know. You're gonna actually be asked to regurgitate this stuff, okay? So you might wanna know that. It's fairly simple. What do you have to have? Gotta have a receptor, gotta have a connection, so there's gonna be a neuron. The neuron has to have some interchange with something else, that's the interneuron, and then it has to go to a motor neuron, and the motor neuron is all about telling, do this, getting a signal down, and it has to get a signal to what? Well, to something that's gonna be able to form a response. So let's go back here. Here's the sensory neuron the sensory receptor has to be down here, distal to this point right here. It's connected to the sensory neuron, okay? Are we good at that? A little button has to be pushed, a little something has to be taken care of over here, and it sends a signal along the neuron. Here's the interneuron in here. Interneuron then is gonna communicate with the motor neuron, and the motor neuron goes out here to something that, a muscle, or some kind of response that says, hey, pull that arm out of the way, get that hand off that hot grill. Okay, so what we did was we just looked at this pathway here. A receptor, it's not shown on that illustration, but we did see the sensory neuron, we saw the interneuron within the spinal cord, we saw the motor neuron originates within the cord, and then the effector organ is going to be a response to whatever the sensory receptor gave as a signal. Now, based on what I told you just a few moments ago, is it always necessary that you have to respond to that reflex there? Summertime, nice hot day. You put your hand down on the car seat, just feels, or maybe the steering wheel or something like that, it's just really hot and it says, oh, that feels so good. Or, hey, that's hot. I think I have a permanent head of Lincoln on the back of my thigh right here. I was in Phoenix, Arizona. It was 113 degrees. Had the car, the windows rolled up on the car. And there was a penny on the car seat, and I got in the car seat and I sat down, and then it's just like I felt like I got branded. It was just like you could hear the flesh go. You know, when we put the steak on the hot grill. I got up so fast, and I says, "Do I have Lincoln's face on my leg here?" Sure felt like it. Man, that was hot. Or I could have just sat there and says, "Oh, that feels so good." Do you have to withdraw? It wasn't that I had to withdraw, but I was getting a signal that this is really uncomfortable. Do you follow what I'm talking about? So I have a choice. In this particular case, I could override the reflex, or I could allow the reflex to take place. Do you follow what I'm talking about? Okay, so reflexes are given there. The point here is it says homeostasis. Automatic response to a stimulus that occurs without conscious thought. So there's homeostasis involved there, it's trying to protect you. But if you choose to do it or not. But homeostasis, in many cases, it is responding for the purpose of making sure that the body is safe or stable here. We're gonna have classifications. We have monosynaptic and polysynaptic. We'll deal with those in a minute. The innate reflexes. Innate. Did we talk about that back in day one or something like that? We made reference to innate? Maybe not. Innate. Inborn. Does anybody ever put the finger in the hand of an infant? Put your finger in the palm of an infant. What do? They got a clutch reflex, right? They do that. How does a child sample their world? Everything goes into their mouth, right? They taste, I mean, it's a moral reflex. They're suckling, a suckling reflex. The baby is just born, and they bring it up to the mother's breast, and what does the child immediately start doing? They start suckling. It's a moral reflex. It's a natural inborn reflex, survival type of thing like that. It's a reflexive. That's innate. They didn't have to teach that kid. We're gonna go to school, learn how to suckle, kid. No. There's an inborn reflex that takes place. Normal, part of a normal response. The startle reflex. They do this oftentimes in, the baby well care, take the child and they have them about an inch or so off the table and they drop them. What does the child do? Does this startle reflex, okay? No. The most fun is you hold them about 3 feet up and then you drop. No, don't do it. Did I say that? I did. Okay, but it's just a little startle reflex. You just let them drop a little bit and they get startled like that. Oh, it's a normal reflex, okay? Then there are learned reflexes. Do you have some? Bus is coming, get out of the way, dummy. Okay? Otherwise you're a hood ornament. You see that little Mercedes-Benz emblem on the side of the kid's body? That was when you got hit by the car. Okay? A learned reflex. There are some reflexes. Have all of you ever ridden a bike? Has everybody ever ridden a bike? Yes?
Mhm.
How long did it take you? Don't answer that. Some of you are still trying. Still working on it. Any skateboarders in the group? There's a dangerous activity for me, at least at my age, maybe if I was younger, but does that take some reflexes in order to stay upright on it? The craziest one I saw was in our neighborhood. There's a little bit of a downhill pitch like this. This girl's on rollerblades, and she's got her dog pulling her. She's got a drink in this hand, she's got the dog leash in this hand, she's on her rollerblades, no helmet, no nothing. She reaches down and she picks up the dog as she's going down this incline right here. Didn't crash, no collision, nothing. I'm just thinking, I got my phone out ready to dial 911. I think this is gonna be fun to watch. I couldn't believe it. I mean, it was just one of these little toy dog type of things, but nonetheless, it's running in front of her, and she's just going down. She's going faster. As a matter of fact, the leash is getting a little loose, and so she reaches down, picks up the dog drink in a hand, and the dog in this arm. No collision. I thought I was going to be able to call 911. Is that a learned type of activity? Holy smokes, that was all kinds of, talk about thalamus? Thalamus was dealing with all kinds of important. Balance, coordination, visual, tactile, all of those things going on at one time. I wish I could have videotaped it. It was really marvelous. It was really, downhill, rollerblades, drink in one hand, scoop a dog. Lady should be on Circus Olay. Somatic reflexes, those are, remember soma? Skeletal, autonomic reflexes, visceral, autonomic. Smooth muscle, cardiac muscle glands. For most cases, the autonomic reflex is a good thing they're autonomic or automatic. We might say, autonomic, why don't you just pencil in automatic? We don't have to think about it. Fortunately, we don't have to think about it because we'd mess them up. Jeez, I heartbeat again. You know? That's the third time this, past hour here. Okay, based on location of the integration center, are they cranial reflexes? Had a few of those in the cranial nerve areas. Are they spinal reflexes? Do they have to associate with the spinal cord here? All of this reflex art stuff right here, we're gonna cover in the lab, okay? So we're gonna cut this off right now. We're gonna go to the next chapter information, okay? Because the rest of this information is, to a certain extent, we're not gonna cover all of it, to a certain extent, most of that's gonna be covered in your, first lab is nerve system, second lab is reflexes. So, what, 3 weeks from now? I think it's next week is the test, and then, okay, you got it there, Sean.